Our next speaker will be Nigel Atkinson.
Well, I'd like to thank the organizers for giving me the opportunity to tell you about some of the work in my lab. Today I'm going to be primarily talking about the work of Alfredo Getzi, who's a research associate in my laboratory. We model alcoholism in the fly, and a fundamental question that we're interested in is how does the central nervous system adapt to alcohol? One of the adaptations that we study is functional tolerance. Tolerance is defined as alcohol-induced alcohol resistance, and functional tolerance means that the tissue or the animal has a reduced response to a given dose of alcohol. The adult fly is ideal for studying this because it doesn't acquire metabolic tolerance, and tolerance in the fly is a neural phenotype. So at the top of the slide is shown how we both induce and assay tolerance. It's a 2-day protocol. We take two groups of age and sex-matched flies. One group is sedated with alcohol vapor, and the other group is mock sedated. And then after recovery, both are returned to food for 24 hours, and the next day we sedate them in tandem, and in this particular assay, we monitor their recovery. And you can see that when we sedate them on day 2, that they sedate very quickly because the dose is so high, and then we immediately switch over to a fresh air environment and they start to recover. And the animals recovering from their second dose recover much more quickly than the animals recovering from their first dose. So this is functional tolerance. We have linked the slow BK-type calcium-activated potassium channel to tolerance in the fly. And the evidence for this has all been published, but I can summarize it as that slow mutations eliminate the capacity to acquire tolerance, that alcohol sedation induces slow gene expression, and that transgenic slow induction phenocopies tolerance, and that the slow expression is required in the nervous system. And so from this and other work, we've concluded that slow induction is important for producing this behavior. Now, what we would like to find is a signature for the alcohol-induced genes so that we can understand how they sense it. We started off by studying the slow gene and characterizing how its histone acetylation time course after alcohol sedation. And in doing this, we've been able to map where transcription factors are acting, and we've mapped, and this helped us show that CREB transcription factor is directly involved in inducing slow after sedation, and we've also mapped a DNA element that has a heat shock factor in. This worked very well for the slow promoter region, so then we turned our attention to the whole genome. Now, the problem all alcohol research labs have is that when you turn to GWAS, genomic studies, and you look at effects of alcohols, you get too many genes responding. It's not unusual to see that 10% of the genome changes expression after an alcohol treatment. And this is seen both in flies and mammals. So to try to sort this problem out, to winnow the chaff from, I guess the other way, the wheat from the chaff, we want to identify the ones that are involved in this particular behavior. And so we used a trick that we discovered quite a number of years ago, and based on an observation that benzyl alcohol and ethanol induce mutual cross-tolerance, suggesting that they are doing so by the same underlying mechanism. We genetically showed that this was the case, that they are dependent on the same genes. Now, these two drugs have very different side effects, and so at the time we thought this would be a good way to focus in just on the genes that are responsible for tolerance. And when we looked at our genomic study, we looked at the entire genome's response and we compared it, both benzyl alcohol and ethanol. We ended up with just a small number of genes in the intersection, 144 genes. And when we organized these using some of Trudy McKay's stocks that are on analyses that are on mod encode, we organized them into co-regulated sets, and it appeared to be working quite well. Now, I'm not going to talk at length about this, but I'm going to come back to this set of data a little bit later. This data, though, to show you an example of it, at the synapse, we ended up finding a number of neural genes. All of the colored ones we've tested by RNAi or mutation or overexpression. sometimes all of them. And what we see is that this assay works very well. This intersection has about an 80% plus hit rate for identifying genes that are involved in tolerance. And you can see that the genes are very closely related. Here we have the para-voltage-gated sodium channel, a beta subunit to the channel, and Pumilio, which is an activity-dependent regulator of para gene expression. And the other genes have also been shown to be genetically interacting as well. Now, one of the other things that we noticed was a lot of the genes that we call tolerance mutants are also well known as circadian rhythm mutants. And this includes the slow gene, the BK channel, CREB, CBP, which I haven't shown you the data for, but we've worked on, and various clock genes that we've shown that clock, timeless, and cycle all interfere with the acquisition of tolerance. And so we thought that perhaps this wasn't a coincidence, that maybe these transcription factors in the last three categories might be important for producing ethanol responses. And so we began by doing a ChIP-seq using a CREB antibody provided to us by Jerry Yin. And this just shows all of the CREB binding sites in the genome, the single spike sites that are mapped adjacent to transcription start sites. The transcription start sites are found about here. And you can see that never sedated animals have a moderate number, but as soon as you ethanol sedate, this is 6 hours after sedation, you see it increases greatly, and it does as it does with benzyl alcohol sedation. Now, when we then looked at the pairing between clock, cycle, and CREB, and this is using data from Mireles Filho in a current biology paper in 2014, what we saw is that there were, in never sedated animals, there's quite a coincidence of CREB and clock cycle sites, but that this particular motif increases in ethanol and benzyl alcohol sedated animals. So one of the things that we've thought is we've looked at this and we looked at the intersection between all of these, the three-way intersection, and this is not drawn to scale, so I apologize for that. But what we saw was that ethanol and benzyl alcohol overlap substantially in their CREB binding sites, and that when we then intersect this with clock and cycle, we see that we also get quite a few intersections. And there aren't as many clock and cycle pairs as one might expect in the genome. There's only actually a moderately small number, I would say. But out of those, 77 of them intersect with the ethanol CREB and benzyl alcohol CREB binding sites. Now, I want to point out that these are sites that are induced by ethanol and benzyl alcohol. These aren't just the static sites that are there as the animal's doing its day-to-day life. So when we look at these 77 genes, we find that they contain most of the well-characterized tolerance genes from the literature. And these references are papers that show that these genes that on the left are involved in tolerance. The ones that don't have references are ones that one would probably predict would, based on other work in the papers, would be involved in tolerance. And you can see that the slow gene falls back in this list. It didn't have to. We didn't force it to belong to this list. And its beta subunit, SLOB, is also in here. And we also see Pumilio returns to this list. And other genes that are like the well-known hangover gene, which is a stress response and alcohol response gene. Perhaps most interesting to me is that out of that 144 genes that we identified in our histone acetylation assay that I mentioned earlier, that 55 of these overlap with this 77. So out of the 77, 55 of them were identified by a completely independent assay, where we assayed just for histone H4 acetylation. So we think that perhaps that this motif is one class of alcohol-induced gene, cycle, clock, and CREB. And we've also since shown that CBP is important for acquiring tolerance. And that is coincident with most of these binding sites. Now, there's still a lot to be done here. We also have quite a few CREB down sites, not as many as we have CREB up. We have about 30 CREB sites where CREB binding is depressed that are also paired with clock and cycle. And then we also have actually a surprisingly large number of clock-only sites where only the clock transcription factor is present and there is no cycle, but it's paired with a CREB site. So I think that perhaps that this might end up being a second type of alcohol-responsive gene, and we haven't studied that yet. We have quite a few clustered CREB sites that aren't near any particular gene. They're not easy to associate with anyone. And one possibility is that these are buffer sites of some sort, that their purpose is to perhaps acquire and pull CREB out of the nucleus to lower the effective concentration to perhaps increase specificity elsewhere. And these are things that we're planning on testing, the buffer sites perhaps by deleting those regions and seeing if this increases CREB binding elsewhere. So I would like to thank the people that did this. This is primarily the work of Alfredo Getzi in my lab. It also has some observations and small bits of data from former students, Harish Krishnan, Xiaolei Li, Yan Wang, Yasha Pol, and Yazan Al-Hassan. And I would like to point out that my graduate student, Annie Park, has a poster on alcohol drinking in the, I guess, tomorrow's poster session. So thank you very much.
Great. Do we have any questions? Can't tell if we're going. Yes? We have a question. We have a question for you.
Sorry. So given that you found this connection to circadian regulators, have you tried going back and doing some of these experiments at different times of day to see if there's any circadian gating on these effects?
So we haven't done that, but the Lyons lab has done that. And what they've seen is that while alcohol resistance cycles, the resistance is innate resistance. It's not induced resistance. It cycles. That tolerance, alcohol-induced alcohol resistance, doesn't cycle. So there seems to be no circadian rhythmicity to tolerance, which I think everybody was surprised about. But thank you.